Please join me in welcoming Dr. Helen Fitzsimons to speak.
Thank you. I'd like to thank the Neurological Foundation for inviting me here to speak today. I was previously overseas for about 7 years, and I was studying the development of treatments for gene therapy of neurological disorders, mostly Parkinson's disease. And I returned to New Zealand 4 years ago after I was very fortunate to receive a repatriation fellowship from the Neurological Foundation, in which I could start studying memory at Massey University. So my research on memory is really at what I consider the real nuts and bolts level of memory research. So I'm interested in how DNA and proteins interact together inside neurons in order to form and store long-term memories. And I use the fruit fly to do this, and I'll explain to you in this talk why I use Drosophila and why I think it's a good model for understanding the real basic fundamentals of how we form and store memories, and also how other small modern organisms can be used in order to understand memory processes. So in order to talk to you about memory, I'm going to give you a little bit of a background on neurons and memory, and explain a little bit about how neurons form and store memories. So memories are formed in neurons, and you can think of the brain as a really complicated big electrical circuit made up of neurons, which are the components. And the neurons are connected to each other, and they communicate through synapses. So you can think of a synapse kind of like an on-off switch. So this here is a picture of 3 neurons connected together. So each neuron has a cell body, and it has a nucleus, and the nucleus is the part of the cell that contains the DNA. So the DNA contains the genetic code, and one of the main purposes of DNA is to provide a template or a blueprint for making protein. So DNA is a code for protein, and proteins are the building blocks of the cell, so it forms the structures to form the outside of the cell, and a lot of the functions within the cell are performed by proteins such as enzymes. You can see these tree-like structures that are sticking out of the cell body. These are called dendrites, and these are the part of the neurons that receive information. And then the cell transmits information to other neurons through these structures here called axons. So if we zoom in on this little region here to this is a picture of an axon. So this is the part of the neuron that's sending information to the dendrite of the following neuron. And the way that our neurons transmit information is through these little chemicals called neurotransmitters. So what happens in this electrical circuit is when this neuron here gets activated, it sends an electrical impulse down the membrane, which causes the neurotransmitter to get released into the synapse. And if sufficient neurotransmitter gets released, it binds to the surface of the dendrite and activates this neuron. And with sufficient activation, it will cause an electrical impulse to go down the membrane of this neuron to the next following neuron. So that continues the circuit. And memories are encoded by interconnected networks of synapses. So in the human brain, there are about 86 billion neurons. And each neuron has, on average, around 7,000 synaptic connections with other neurons. So if you kind of put these two numbers together, there are about 10 to the 14, which is 10 with 13 zeros after it, to 10 to the 15 synapses in an adult brain. So you can see how really complex a human brain is, but also how it can have the capacity to store all these probably hundreds of thousands, if not millions of memories that we form over a lifetime. But trying to understand how we form and store memories within neurons is quite a daunting prospect. When you look at a brain like this, you can think, well, where on earth would you start? And this was the kind of problem that was faced by a neuroscientist called Eric Kandel in the 1960s and 70s. And he was one of the pioneers in the field of trying to help us understand what are the basic processes involved in forming a memory, what happens inside neurons to form a memory. And he thought that the human brain was going to be far too complex, so he thought up a different approach, and his rationale was that he knew that there were many small organisms that had a great capacity for forming memories. If you think of a honeybee, how it goes out and finds pollen, and then it manages to remember where its hive is. It has this really complicated way of coming back all the way to its hive. And there are many other examples of small organisms that have quite complex memories. So Eric Kandel, he formed a hypothesis that because so many organisms can form memories, that the basic molecular pathways involved in forming memories would be ancient and they would be conserved. And if he tried to understand what was happening in a small model organism, then this would be able to be applied to higher order organisms such as mammals. And he chose to use the sea slug Aplysia. So this is Eric Kandel. He's in his 80s now, but he's still working. He's at Columbia University. And this is the sea slug Aplysia. So I guess it's a matter of opinion whether you think it's a beautiful organism, but I think he thought that it was. And when you look at it, you can think, well, how on earth could we ever learn anything from that and apply it back to a human brain? And he said his colleagues thought he was a bit crazy, but he persevered. And the reason he chose Aplysia was because it has a small number of neurons. Still has 20,000, but that's about 4 million times fewer than a human brain. And importantly, it has behaviours that can be modified by learning. So what he aimed to do was to get the slug to learn something and then do biochemical experiments to figure out what was changing inside the neurons at the DNA and protein level and correlate that with learning. And the behaviour that he chose to look at was called the gill reflex withdrawal. So this is the underside of the sea slug. He had to, I think, cut it open a little bit to expose a siphon, which is this here, and a gill, which is in yellow. And he found that, well, I think it was already known, if you touch the siphon, the gill will withdraw. So you see the yellow bit gets a little smaller. But if you give a tail shock, at the same time as you touch the gill, it will touch the siphon, the gill will withdraw a lot further and for longer. And when you come back later on and just touch the siphon, the gill will still withdraw a lot more than it would had it never had the tail shock. So this is a type of sensitization. So Kandel did these experiments and he found that if he came back 10 minutes, if he gave the tail shock and touched the siphon and then came back a number of minutes later and just touched the siphon, the gill would withdraw, and this was a short-term memory and it lasted for up to an hour and then slowly decayed. So he could get the slugs to learn. So then what he did was some biochemical experiments to figure out what was happening inside the cells. And he found that there was a transient increase in a little molecule called cyclic AMP, which I call CAMP for short. And he already knew of this molecule because it had been quite well characterised. It's involved in a number of other cellular processes in the body. And what it does is it activates a protein called protein kinase A. And protein kinase A itself activates a number of proteins in the cell. So what he found was that if this is the neuron that receives information from the siphon, it has a synapse with the neuron that receives the information from the tail shock. And when you give a tail shock and touch the siphon, cyclic AMP increases and it activates protein kinase A, which activates a number of other proteins in the cell, and this results in a transient increase in neurotransmitter. So this is a larger activation of the following cell, and this is connected to the motor neurons that are attached to the gill reflex. The gill reflex. So then when he came back later and just touched the siphon, there was still this transient increase in neurotransmitter release. So that's the molecular basis of what the short-term memory was. So then he thought, well, could I get this slug to form a long-term memory? And he knew that, as with human memory, it requires repetition a lot of the time. The more you repeat something, the easier it is to store it in your long-term memory. So what he did is he touched the siphon and gave a tail shock, and then repeated this 3 more times, and then did this whole training session each day for the next 3 days. And then he came back 24 hours later and just touched the siphon, and the gill was withdrawn much longer than it had previously been. So the slug had formed a long-term memory, and he could come back even 7 days later and he would still have this increased gill withdrawal reflex. So the slug had formed a long-term memory. And what he found was that the initiation of the process was the same as for short-term memory. There was this increase in cyclic AMP, which activated protein kinase A. But then what happened was when there was so much protein kinase A activated, it actually moved into the nucleus of the cell and ordered proteins to be made. So it activated the DNA code for making proteins, and these were specific proteins involved in growing new synapses. So these synaptic proteins get incorporated into the synapse, and it grows bigger. And this means it can release more neurotransmitter, and this is a sustained effect lasting up to at least 7 days. So in summary, long-term memory requires repetition, and it requires synthesis of new proteins, and these are specific synaptic proteins involved in growing bigger synapses. And I think not only just increasing the size of the synapse, but actually growing new synapses from the same cell as well. So around the same time that Eric Kandel was doing these experiments, there was another researcher also in the United States called Seymour Benzer. And he was also interested in the neurobiology of behaviour, and like Eric Kandel, he was also a reductionist. So he believed that memory could be broken down into individual interactions between proteins and cells. But he took quite a different approach, which was to use the fruit fly Drosophila to try and understand memory. And he knew that he could train fruit flies to form and store memories, and he used an assay called olfactory conditioning, where he could get the flies to associate an odour with an electric shock. So he'd expose them to an odour, give them a shock, and then later on when he came back and exposed them to the odour, they would avoid it because they were associating it with electric shock, even though the odour itself was not harmful. So then what he did is he treated thousands of flies with a chemical called ethylmethane sulfonate, which is a DNA mutagen. So it will mutate single base pairs of DNA, which will cause, these mutations will cause proteins to not be synthesized correctly, or some genes there will be no protein made. And he took these flies and tested them in his olfactory conditioning assay, and he found several mutants that had deficits in memory. And then he spent about 10 years trying to figure out where in the genome these mutations were, so which genes were mutated so that they weren't producing the proper protein. Nowadays it's quite easy for us to do these kind of studies, but back then it took many years, so it took a lot of perseverance. But lo and behold, the first 2 genes that he was able to characterise and find the mutations in, the first one was rutabaga, which codes for a protein called adenyl cyclase, and this is the enzyme that catalyses the synthesis of cyclic AMP. And the other protein was called, gene was called DUNS, and this codes for a protein called CAMP phosphodiesterase, which is involved in breaking down cyclic AMP. So both of these genes were involved in the cyclic AMP signalling cascade, and this was quite exciting because even though a sea slug and a fruit fly are hundreds of millions of years evolutionarily distant from each other, the same basic biological pathways are involved in forming and storing memories. So Eric Kandel's hypothesis was correct. We know from a lot of studies since this time the CAMP pathway is important for memory in many different organisms, including mammals. And he received the Nobel Prize for Medicine in the year 2000 for his work on aplysia. So the research, since this time we know a lot more about the specific proteins that are involved in forming synapses and forming long-term memories and how they interact together, but we still have a lot of questions. And one of the things that I'm interested in is the regulation of memory. So there are quite a lot of constraints on the memory system, because if you think about it, remembering is important, but forgetting is also important too. We don't want to form long-term memories of everything. You don't care what you had for dinner, 13th of March 2007. Like, there's a lot of things that aren't important. And you can probably all think of examples of humans who have amazing capacities for specific types of memory, amazing memory formation in some circumstances. So the brain normally constrains this, and I'm interested in how this regulation is occurring. How do we prevent memories from forming? Because if we can understand that, then if we could inhibit that system and maybe enhance memory, this could be we could use this for new approaches for development of therapeutics to improve memory for individuals with cognitive disorders. So in order to explain my research to you, I need to introduce some key concepts. My research is on the role of histone deacetylases in memory, and their catalytic activity is on chromatin. So I need to explain chromatin, what we know about histone acetylation in memory, the use of Drosophila as a model for memory research, the assay that I use to measure memory, which is the Courtship suppression assay, and then some information about what I'm currently doing in the lab and the future directions. So what is chromatin? Well, inside all of your cells, you have a nucleus, and if you took the DNA in the nucleus and spread it out into one long strand, it would be about 1 metre long. But the diameter of each nucleus is only about 10 nanometres. So the DNA needs to be compacted about 100,000-fold to fit it into the nucleus. And the way that it does this is to be packaged into chromatin by interacting with proteins called histones. So this is a strand of DNA, and it gets wound around these orange structures, which is kind of a circular depiction of what a complex of histone proteins looks like. And we call this the beads on a string form of chromatin. And this gets wound around itself again and again to form these higher order structures until you end up with a chromosome. So histones and DNA interact together to form chromosomes, but chromatin is also really important for regulating gene expression. And by that I mean determining when and where DNA is used as a code to make proteins, because it's really important that we make the right proteins in the right cells at the right time. And one of the ways that gene expression is regulated is through histone acetylation. So this is your DNA wrapped around the histone proteins, and right now it's in a closed chromatin structure. And when it's in this closed structure, it can't be used as a template for making proteins. It needs to be opened up a little bit to allow specific regulatory proteins to be able to come in and read the DNA code in order to synthesize proteins. And one mechanism for doing this is through histone acetylation, and this is through the action of enzymes called Histone Acetyltransferases. And they add this little thing we call a histone group onto a histone, which changes its charge, and it releases slightly from the DNA, so it unwinds a little bit. So this is an acetyl mark. The histones unwind a little bit and allow access to the regulatory proteins, which allow gene transcription, which is another way of saying making proteins. And the histone acetylation can be reversed by the action of histone deacetylases, which will take off the acetylation and close up the chromatin so that you can't get gene expression. And to illustrate why this is important, you can think of how does a cell remember who it is. So in your body you have lots of different types of cells, and say in your heart you have specific genes that make heart proteins. But you don't want the genes that make stomach proteins to be active in your heart, because if you imagine making digestive enzymes in your heart, that wouldn't be a good thing. So during development, certain genes get turned on and off. So heart-specific genes stay on and stomach-specific genes get turned off in the heart. And this is through deacetylation of histones to close up the chromatin structure, and this is permanently maintained during your life. And the reason that I'm interested in histone acetylation is because it correlates with memory formation. So in the rat brain, if you train them in a learning test, like a maze or something like that, there is an increase in histone acetylation, and this is in specific areas of the brain that are known to be involved in memory formation, such as the hippocampus. Loss of histone acetylation impairs long-term memory formation, and an example of this is a mutation in a histone acetyltransferase if you can't get any histone acetylation occurring. In treatment with histone deacetylase inhibitors, which overall increases acetylation, can improve long-term memory in rodent models. And lastly, histone acetylation is abnormal in rodent models of Alzheimer's disease. So this increase in histone acetylation that normally occurs during learning doesn't occur in some of these models of Alzheimer's disease. So these data taken together suggest that histone deacetylase inhibitors may be negative regulators of memory. So if you can develop specific drugs that inhibit histone deacetylases, we might be able to improve memory. But at the time that I started this research, little was known about which specific histone deacetylases are important for histone acetylation during memory formation. And in humans, there are 11 of them. So my research questions were to figure out which histone deacetylases are important for regulating memory formation and how are they working, how are they regulated. So my initial strategy was to manipulate the amount of each histone deacetylase in the adult brain, and I'll explain to you soon why I took this approach, and determine the effect that this had on long and short-term memory. So then I could understand which ones were involved in memory and study those ones further. So I used Drosophila as my model for studying memory. It's just a little fruit fly that buzzes around your bananas. But actually, it's been a very valuable tool in scientific research for the last 100 years or so. What we know about chromosome segregation was initially elucidated in Drosophila, and it's most well known for its role in helping us understand embryo development. So, and how embryos, pattern and form limbs and that kind of things. Because if you look at a little fly, it has a head, it has a thorax, it has a chest, an abdomen, legs, it has internal organs, so in some ways it's not that different from us. And the biological properties involved in forming those structures are well conserved through the whole animal kingdom. So it shares many essential biological properties with humans. Its genome is very similar to humans. It has 13,500 genes. Humans have about 20,000, so even though we're much bigger, we're actually not that much more genetically complex than a fruit fly. And a lot of the extra genes that humans have are from duplication events. So Drosophila might have one copy of a specific gene, and it will have duplicated and formed slightly different functions, two slightly different functions in humans. They're really great for genetic analysis, which I'll get to in a minute. You may be surprised that they have quite a complex brain. It's still very small, but it has 200,000 neurons, and it's a highly organised, ordered structure. And some researchers wrote a big list of all the human disease genes that they could think of and compared this to the Drosophila genome and found that about 75% of them were conserved. But importantly for memory research, Drosophila displays reproducible behaviours that can be modified by learning. So we can treat Drosophila with drugs or alter which proteins it makes and then determine what effect this has on memory to try and understand which genes and proteins are important for memory formation. Memory-related genes are conserved in Drosophila. I already talked about the CAMP signalling cascade. These are the names of the Drosophila genes. Drosophila researchers think that they're quite funny and they always come up with strange names for their genes. A lot of genes involved in growing synapses and synaptic function, and also diseases involved in cognition. So this gene is involved in Alzheimer's disease, Fragile X syndrome, Down syndrome. So why do I use Drosophila? It's mainly for the genetic analysis. It's very easy to make transgenic flies. And I can also order mutants in almost every gene from a stock centre. So if you're interested in a specific biological function, you can order a whole lot of mutants and test that function in all of the mutants, and any that don't have an impaired function, then you can figure out what gene it is that's causing that is involved in that function. And I can also increase or decrease expression of almost any gene in almost any tissue at any time of development. And to illustrate how useful this can be, I use the eye as an example. So there's a gene called PAX6, and it's involved in eye development. So this is a normal Drosophila head with mouth parts and these large compound eyes. If you decrease the amount of PAX6, like you take a fly with a mutation in the gene so it doesn't make the protein, it doesn't form eyes. So that proves that PAX6 is involved in eye development. You can make a transgenic fly where you put the PAX6 gene back in and you can reform the eye. You can also increase the amount of PAX6, and some researchers took the PAX6 gene and they increased the amount of it in the antenna. And you may wonder why on earth would you want to do that? That seems kind of silly. But it actually had a really important, there was an important reason for doing it, because right on the end of the antenna, you might be able to see that there's a little primitive eye that's been formed. So this tells us not only is PAX6 the gene that's really important for developing the eye, but it's also a master regulator of eye development. It starts off the eye development cascade. And this data can also be applied to human health because PAX6 is involved in eye development in humans, and humans that only have one copy of the PAX6 gene have blindness with this disease called aniridia. So we can study eye development in Drosophila by increasing or decreasing the amount of a specific protein and try and understand the genes and proteins involved in forming eyes, and then this may be helpful for understanding eye development in humans. So I need to explain to you a little bit about the Drosophila brain. There's a big head here. It has mouth parts, antennae, these large compound eyes, and inside this head capsule is quite a big brain. And I'm interested in the part of the brain that is in these red L-shaped structures here, and it's called the mushroom body. And the mushroom body is quite similar to the hippocampus of a rat or mouse or a human. It's the part of the brain that seems to be the most important for memory formation. And the evidence for this is that if you chemically ablate the mushroom body, so you basically kill it so it can't function, the flies can't learn. Mutants that don't develop proper mushroom bodies also can't learn. And if you inhibit neurotransmission from the mushroom body, that is, you prevent the mushroom body from being able to send information to the following neurons in the electrical pathway, the flies also don't learn. And a lot of the genes and proteins that are known to be important for memory formation tend to be concentrated in these mushroom body neurons. So this is the mushroom body up close. It's a bilateral structure, and on this image it's slightly rotated. So here are the L-shaped lobes, and they're at the anterior part of the brain, so they're right at the front. The cell bodies and nuclei of the mushroom body neurons are all clustered together in these circles at the back of the brain. And they send their dendrites, the part of the neuron that receives information, into this little area here called the calyx. And they receive information from these green neurons, which are the antennal lobe neurons. They come from the antennae, and they're a part of the olfactory system, because olfaction is really important for memory in Drosophila, because they rely a lot on their sense of smell. And then the axons, the part of the neuron that sends information to other parts of the brain, they are all clustered together into this fibre called the peduncle, and they form these lobe structures here, and they send information to other parts of the brain. So in order to measure memory in Drosophila, I use the Courtship conditioning assay. So this relies on the premise that a male fly remembers that he's been previously rejected by a female. So in that way, they're not that different from humans either. So in Drosophila, the female will only mate one time, and then she will actively reject a male. So evolutionarily, it's in the best interest of males that they learn what rejection behaviour is, so that they don't bother wasting their time courting females that they're never going to be able to mate. So what I do is I take a male, which is here, and he's of a genotype, so he's a certain gene changes that I'm interested in. And when he closes from his pupal case as an adult, he is housed individually or by himself. So he never sees an adult female until I put a previously mated female in this little mating chamber with him. So the first time he ever sees a female, she actively rejects him. And over the next 7 hours, he will have multiple bouts of courtship behaviour where he'll try really hard to court her, but she'll reject him. And these multiple bouts with breaks in between represent the repetition required for formation of long-term memories. I also have a control male, who I call the sham, and he's all by himself. He never gets to see a female. So after 7 hours, I take out this female, and then I wait 24 hours, which is the time frame for long-term memory in Drosophila, and then place each male with a new freshly mated female, and I measure courtship behaviour over the next 10 minutes. And the males that have learnt what rejection behaviour is and not to bother courting her will display less courtship activity. And the behaviours that I'm looking at are orienting. So these are the males with the darker abdomens. He sits at the specific angle towards the female where she can see him, and she knows he's interested. He'll tap her. He'll sing to her. He puts his wing out and flaps it, and it vibrates and she can hear it, and we call that the love song. He'll taste her pheromones, and he'll try to mate her, but he will never mate her. No pornography. So this is an example. I'm sorry about the quality of this movie. So there's the male chasing the female. He's got his wing out, he's vibrating it, she can hear it, but she's rejecting him. This is positioning, he's sitting in the spot, and he's getting ready, he does his love song, he tries to mate her, but she won't let him, and she runs away. And it keeps going on and on like that. So then from that, I calculate a memory index. So for each group of flies, I test at least 20 flies, because like humans, they have variations in their behaviour. And I measure the courtship activity of a sham male, so this is the proportion of time he spends over 10 minutes in courtship activity. So he spends about half his time trying to court the female, where a trained male who's learned not to bother will only spend about 25% of his time in courtship behaviour. So then I get each score for each of these males and divide it by the mean of the sham group to come up with a memory index. So at this point, all you need to remember is the higher the bar, the worse the memory. So if the bar is up near 1, this means that a trained male spent as much time as the sham male in courtship activity, so he hasn't learned anything. Between about 0.4 to 0.6 is a normal long-term memory score for a normal fly. I can also measure short-term memory, where I train the flies for just 1 hour and then I measure their courtship activity straight after. And that score is usually less than 0.4 because it's a stronger memory. So what I want to do is to genetically manipulate the level of each histone deacetylase in Drosophila and determine the effect on memory. So in humans there are 11 histone deacetylases, and in Drosophila there are 5. And the ones that I'm going to talk to you today about are RPD3, which is most similar to human HDAC1 and HDAC2, and HDAC4, which is most similar to the class 2A, HDAC4 and 5 in humans. So about a year ago I finished up some work looking at RPD3, and I found that RPD3 is really critical for forming long-term memories in Drosophila. When I increased the amount of RPD3, it caused a specific impairment in long-term memory, but short-term memory was normal. And if you remember that long-term memory requires proteins to be made, it requires protein synthesis. And histone deacetylases regulate the DNA such that it can or can't synthesize proteins. So it makes sense that the action of RPD3 affects long-term memory, but not short-term memory, which is completely independent of protein synthesis. And there have also been 2 papers that have been published on the mammalian forms HDAC1 and HDAC2. And these 2 papers were published in the scientific journal Nature, which is probably one of the 2 or 3 most prestigious scientific journals that there are. It's from the same research group that were at Harvard and MIT, and they're quite beautiful papers. And they found similar results, which was that overexpression, so increased HDAC2 protein, impaired long-term memory, had no effect on short-term memory, and they found no effect with HDAC1. They also showed that if they decreased the amount of HDAC2, they got an enhancement of memory, and there was an increased amount of synaptic proteins that were made. So now long-term memory depends on protein synthesis to increase the amount of these proteins that get made into new synapses. They had increased amounts of these proteins being made. And they also found that in mouse models of Alzheimer's disease, there was increases in HDAC2 in their brain. They also looked at human Alzheimer's disease patient brains compared to non-Alzheimer's brains, and also saw an increase in HDAC2. So taken together, these data suggest that specific HDAC2 inhibitors might be a promising target for drug development, because so now we've identified a specific histone deacetylase that impairs memory. It's not an easy task because we don't want to impair HDAC1, because there are other studies showing that HDAC1 is important for protecting neurons from cell stress, so you don't really want to decrease that. So there's a lot of work being done on development of specific HDAC2 inhibitors, so maybe in the future one day that there'll be some new drugs developed that might be quite helpful for cognitive function. Today I want to talk to you about some new research that I'm just starting on HDAC4. And I started thinking that HDAC4 might be involved in memory formation because, well, firstly it's expressed in the adult mammalian brain in the hippocampus, and I also found that it's expressed quite strongly in the Drosophila mushroom body. There is also a human disease called brachydactyly mental retardation, which in some cases appears to be caused or correlates with a mutation in the HDAC4 gene. And the symptoms of this disease are impaired learning and behavioural problems and autism. Also, interestingly, with HDAC4, it's also expressed at synapses. So if this is a picture of a neuron, here's its nucleus, here's its dendrites, and an axon. Histone deacetylases you'd think should be in the nucleus because that's where the histones are. But actually, a lot of HDAC4 protein is found outside the nucleus. There's some in the cell body around the nucleus, and there's also some in the dendrites and in the axon. And actually, in the mouse brain, there's a pool of concentrated HDAC4 right at the synapse. And normally in cells, if a protein is concentrated in a specific area, it has some kind of function in that area. So HDAC4 might also have a synaptic function. And when the neurons get activated, such as during memory formation, HDAC4 gets shuttled out of the nucleus. So if you hypothesize that HDAC4 was also a negative regulator of memory, when the neuron wants to form a memory, it might make sense that it kicks it out of the nucleus, and possibly also maybe it has a pro-memory function in the synapse. So this is my hypothesis and why I wanted to look at this protein more. One of the known kind of characterized functions of HDAC4 is in muscle development, where it interacts with a protein called MEF2. And MEF2 turns on genes involved in muscle development, and HDAC4 inhibits MEF2, so it can't do that. And I also already knew that MEF2 is also expressed quite strongly in the mammalian hippocampus and the Drosophila mushroom body, so I want to examine this interaction as well. So my research questions are, what role does HDAC4 play in memory formation, if it does play a role? What proteins does it interact with, so how is it regulated? And also, like HDAC2, is it also altered in human Alzheimer's disease brain? So the first thing that I wanted to do was to genetically alter the amount of HDAC4, so increase it or decrease it, and see what effect this has on memory function. So I can genetically manipulate the fly so that they have an increased amount of HDAC4 in the mushroom body. And in order to check that it is expressed in the mushroom body in the right place at the right time, I need to be able to have a look at the brains. So in order to look at HDAC4, I engineered the flies to have a little tag on the end of it called a flag tag. And I have an antibody to a flag tag that I can hook up to a fluorescent probe. So then I can use microscopy with a fluorescent detection to see exactly where in the brain the HDAC4 is. So a Drosophila brain is about 0.5 millimetre long. So this is my PhD student Sylvia dissecting fly brains. You need to use a microscope and very fine forceps, and it takes a bit of practice. And I was trying to think of an analogy of like what it's like to dissect a fly brain, and the best thing I could think of was it's like using barbecue tongs to pull the skin off a mandarin. Like, you can do it, but it takes practice. So these are what the little brains look like in a dish. And then in order to look at the fluorescence, we use a confocal microscope. And this is a pretty cool tool where it can take optical sections, it can scan right through the brain so that you can then get a 3D picture of where the fluorescence is. So this is overexpression, increasing HDAC4. So this is half a brain I'm showing here, and I have an antibody which is showing in green to RPD3. So this is the other histone deacetylase that I worked on, and it's only in the nuclei. So this shows basically the outline of the brain where all the nuclei are. And these black spots are just areas of the brain where the nuclei aren't clustered, and that's where like dendrites and axons are and things. So HDAC4 is in here. This is from the anterior side of the brain, so I can see the lobes, the axons, and I can see strong HDAC4 expression. So you can immediately see it's not just in the nuclei, it's actually in the axons. And then if I have to flip over the brain to the other side to see the cell bodies in the nuclei, because they're at the back of the brain. So again, this is RPD3 showing the outline of the back of the brain, and this is the HDAC4. So this is the circular region where the cell bodies and nuclei of the mushroom body are. And we can take an optical slice right through the middle of that area and see it in more detail. So this merged column here is where the HDAC4 and the RPD3 lanes are merged together, and any time that they co-localise, you'll see in yellow. So you can see that RPD3 is in the nuclei and not in this region in the middle, which is the calyx where the dendrites are, but HDAC4 is in the dendrites. And then to zoom in on this region here where the cell bodies are, you can see RPD3 is in these little circles of nuclei, whereas HDAC4 is kind of in this honeycomb pattern around the cell body. So it's in the cell body but not in the nucleus, except here and there in some cells there are these really concentrated nucleus spots of HDAC4, and they'll become interesting in a little while. So in summary, I can increase the amount of HDAC4, and it seems to be in the axons and in the dendrites and sometimes in the nuclei. So there seems to be some kind of tight regulation of the subcellular distribution of HDAC4. So the next thing I want to do is test it in my memory assay. So I engineered the flies that they increase HDAC4 in adulthood, and then I tested them for 24-hour long-term memory. And these two lanes here, these two bars, are normal flies with normal HDAC4 levels, and they have normal levels of memory, whereas there is a specific impairment in long-term memory when HDAC4 is overexpressed. I also looked at short-term memory, and there were no significant differences. I actually haven't quite finished testing these flies, that's why the error bars are a little big, and probably those numbers will get closer together, but there's clearly no impairment in short-term memory. So this is consistent with HDAC4 probably having a role in regulating protein synthesis, making the proteins required for growing the synapses. So then first thing I wanted to do was look at MEF2, because as I said before, it is involved in muscle development and interacts with HDAC4, and I know it's expressed in the mushroom body, so potentially one of the mechanisms through which HDAC4 could be acting is regulation of MEF2. So I looked at the expression of MEF2 in fly brains, and it's also a nuclear protein, so it's here in all the mushroom body nuclei. And if we zoom in on one of the nuclei, you can see here that the flag is in the area of the cell body that's not the nucleus, but any time that there are these little concentrated spots of HDAC4 expression, it also co-localizes with MEF2. So they're in the exact same spot in the nucleus, so it's quite likely that they're interacting together. So now what I'm doing is examining the role of MEF2 in long-term memory and whether this is interacting with HDAC4 and they're forming some kind of memory network. I also decreased the amount of HDAC4 in the brain to examine what effect that had on long-term memory. And I also saw a specific deficit in long-term memory. So you may think this is a little bit curious. When you increase the amount of HDAC4, it impairs memory, but also when you decrease the amount, it also impairs memory. So too much of it is bad, but you need some of it there. Oops, I'll go back. In the last 6 months or so, when I was doing this research, there were absolutely no publications on HDAC4 and long-term memory, but in the last 6 months there have been 2 papers which are very interesting because they're both in mouse models, and in one of them they increased HDAC4 in the hippocampus, but they had a specific mutant that was only nuclear, so it can't go in the rest of the neuron. And this caused a specific impairment in long-term memory but not short-term memory, so that's similar to what I saw. And there's also another paper where they made a brain-specific knockout of HDAC4, so they decreased HDAC4 in the whole brain. Actually, it might have been just in the hippocampus, but there was no HDAC4 there, and this also caused an impairment in long-term memory. So what I think is happening is that when it's in the nucleus, HDAC4 is actively inhibiting memory by probably inhibiting the expression of the genes involved in growing new synapses, but it's important for memory in the cytoplasm, the area outside of the nucleus, and possibly at the synapse since it seems to be concentrated there. So maybe it has a pro-memory effect outside of the nucleus. So my next steps are to try and identify genes that are involved in this HDAC4 memory pathway, how it's working, and I'm doing a couple of screens. So Drosophila is really good at screening, like pulling out what genes are important in certain processes. And so one of the screens I'm doing is looking for genes that alter the subcellular localization of HDAC4. So which genes cause it to go into the nucleus, or which genes cause it to be concentrated at synapses, to try and understand these interactions better. And I'm also using a technique called RNA sequencing, where I sequence there's an intermediate between DNA and proteins, and I can measure the amount of all of the genes that are being made into proteins in one go, and I can see which ones are changed if I increase or decrease the amount of HDAC4. So then I'll be able to identify which genes is HDAC4 altering. I'm also collaborating with Debbie Young at the Centre for Brain Research at the University of Auckland to try and apply the results I get from these first 3 studies to vertebrate memory. So how does the HDAC4 memory pathway work in rodent models of memory, which is more complex memory forms? And I'm also collaborating with Mike Dragunov and his postdoctoral fellow Pratika Narayan also at the Centre for Brain Research. And they have tissue samples from patients that had Alzheimer's disease and also non-Alzheimer's disease patients that are part of the Neurological Foundation brain bank at the University of Auckland, and they've made these little arrays which have spots of tissue from about 30 patients, and they can look on those arrays to see if HDAC4 is altered in Alzheimer's disease. So, acknowledgement. I'd like to thank the funding bodies, Neurological Foundation for the Repatriation Fellowship that allowed me to get a foothold on this research in New Zealand. Then I was lucky enough to be awarded what was then a forced fellowship. I think now they're called MOBI, but those fellowships don't exist anymore, sadly. And I'm now funded by the Health Research Council. And I'd like to thank Jinyu Chen, who helped with the confocal microscopy. I had 2 summer students, Fiona and Melissa, who helped me make some of these DNA constructs. My previous postdoc supervisor, who's now at North Carolina State, is Max Scott. And my collaborators at Auckland, Debbie Young, Mike Dragunov, Richard Fore, and Pratika Narayan. Thank you.
Thank you very much. Dr. Fitzsimons, are you happy to answer a few questions if there are some? Oh, could I just ask you to wait for the microphone, then everyone can hear the question.
Are there different types of fruit flies?
There are several different species, yes. The one that I use is called Drosophila melanogaster, which is, I use a lab strain of that. It's been in research labs for about 100 years. And I think if it actually got released into the environment, it probably wouldn't do very well because it doesn't have many evolutionary constraints on it, being sitting in its food all day. But there are several other variants, and the ones that you see on your bananas, I have a look at them and they're slightly bigger and they have slightly browner eyes, so I think they're a different species.
You've been working on the mechanisms of the formation of long-term memory. Do you think those are the same mechanisms for the retention of long-term memory?
I think it's, when we initially started this research, what we were hoping was that the histone modifications would cause permanent changes to the chromatin that would permanently turn on these genes involved in synaptic function, and the memories would be retained that way. In reality, I think it's a bit more complex than that. Some of these changes in histone acetylation are actually transient. They go up and down rather than being permanent. And the retention of memory, it's quite complicated in that if you have one cell body on a neuron, it has all these thousands of synapses, and it needs to coordinate which synapses are involved in which memory. And there's a theory called synaptic tagging that once a memory is formed, there are signals that are sent between the synapse and the nucleus that cause these synapses to, what they do is they capture the proteins and hold them there so that synapse stays bigger, whereas other ones don't. And that seems to be a mechanism possibly involved in retention of memories, but it's still not very clear. But I do wonder if HDAC4 might have a role in that process, since it seems to also be concentrated at the synapse. So, there's got to be a lot of communication between the cell body and the synapse.
I'm wondering how you fit 7,000 synapses on an axon when they're all at the end, and whether acetylation of histones is preserved through cell division, And chemically, how similar neurotransmitters are to histones?
Well, neurotransmitters are quite different than histones. The neurotransmitter is at the synapse and the histones are inside the nucleus. What's the other question? 7,000 synapses. Well, neurons are incredibly complex. This was just a cartoon of a very, very simple one, but there are many different types of neuron in the brain, and some of them have zillions more dendrites, some have more axons. There's lots of different forms of them. There was one other question. Yes, histone acetylation can be is transmitted through cell division in tissues such as that have dividing cells. The brain, most of the cells are not dividing anymore. So once the neuron is formed as an adult neuron, it's maintained like that through its life, and then it will die and it won't be replaced. There are some neurons that will divide in the brain, but most of them aren't.
Any more questions? Oh, thank you very much, Dr. Simons. You've done a fabulous job in sharing all your research with us, so thank you. And I think we've got to thank all our speakers today because they've really had some incredible insights for us into their work, and they've taken time out of their busy day to come and see us, so thank you. Members of the audience, thank you for attending today. If you didn't register when you arrived and you'd like to receive updates from the Neurological Foundation to events such as this, would you please ensure that you do register before you leave? And we'd like to say a special thanks also to the Victoria University of Wellington and to our volunteers all wearing the I Have Research on the Brain t-shirts today because they've been of great help in running the event. We're just going to ask Dr. Fitzsimons to draw 3 lucky winners for these.
Fun part. All right. Want me to read it out?
Yes, please, if you wouldn't mind.
The first one is Diana Morrison.
Is Diana here still? Oh, we can get that to her because she's left her address with us.
One from underneath. This one is J.L. Cockburn.
Oh, fantastic. We've got someone here. That's lovely. Congratulations.
Big mix
Oh,
around. All right. And this is Oscar Henning Tapley.
Yes. Right. Well, we've got an address for that one too in Mount Victoria. So thank you again. And we hope to bring you more lectures just like this and achievements of brain research in the future. Thank you from the Neurological Foundation.